Now we're going to look at the process of phototransduction. This is how your eyes turn light rays into signals to your brain. So we have here a photoreceptor. So the yellow thing here in this picture is our photoreceptor. We're looking at two different scenarios here. So we have one on each side. The next cell here, this pink cell, is called a bipolar cell. Then the gray one at the bottom is a ganglion cell. The ganglion cell is what goes to the brain. So if you get an action potential here, that will send a signal to the brain. So we have two different scenarios. We're going to have when your photoreceptors are sending a signal to your brain and when they're not. On the left here, we have dark. So this is what your photoreceptors are doing when they are in the dark. And on the right, we're going to have light. So we have two scenarios, when your photoreceptor is in the dark and when it's in the light. Now, do you recognize these things? This is a sodium gate. This is a potassium gate. Right? This goes back to chapter 12 when we first learned about these. Think about what you know about depolarization and repolarization. The basic process is going to be that you let sodium in. So sodium gates open. Sodium comes in and you depolarize. And we know that when potassium gates open, potassium goes out, and this will repolarize or hyperpolarize. The idea is that sodium raises voltage, potassium lowers voltage. So in the dark, your sodium gates are open, and sodium is coming in, and you raise the voltage. Well, because you're raising the voltage, that causes down here, remember this one? This is your calcium gate. So the calcium gate opens. Calcium comes in, and remember calcium coats the vesicles that contain the neurotransmitters so that you can have exocytosis, and here you secrete neurotransmitter. This is an inhibitory neurotransmitter. So we're going to have IPSP. So because we're having IPSP, that means we lower the voltage on the bipolar cell. Remember, IPSPs prevent an action potential in the next cell. So there is no action potential in the bipolar cell. So that means we have no neurotransmitter here, which means we have no action potential here, which means no signal to the brain. So the eyes, again, they're a little counterintuitive. When you're in the dark, there's no stimulation to the photoreceptor. So in the absence of that stimulation, the photoreceptor secretes neurotransmitter. So this is called dark current. Dark current means that in the absence of light, your photoreceptor has its sodium gates open. So this is the opposite of what we've learned before, of how everything else works. All the other receptors and neurons open their sodium gates in response to a stimulation. But with your photoreceptors, it's the opposite. They open their sodium gates in the absence of stimulation. So this means then that the photoreceptor secretes inhibitory neurotransmitter. And then this inhibitory neurotransmitter is what prevents action potentials in the cells that stimulate the brain. So think about the logic of this, of what's going on. Your photoreceptors, when they are in the dark, they are active, and by being active, they shut down the other cells, the bipolar and the ganglion. And that is what makes you not send a signal to the brain when you're in the dark. Then we have light. In the light, now you have stimulation. The stimulation closes the sodium gate. Sodium stops coming in. Well, in the absence of that sodium going in, your voltage goes down because you do still have potassium in. So now that the voltage is down, you have no neurotransmitter here. In the absence of that inhibitory neurotransmitter, your bipolar cell has an action potential. It secretes neurotransmitter. Now you have an action potential in the ganglion cell, and you do send a signal to the brain. Okay, so light actually turns off your photoreceptor, and you lose that inhibition. There's no IPSP. And in the absence of inhibition, the other two cells will have action potentials and signal the brain. So in the light, you have light stimulates your photoreceptor. And because that photoreceptor is being stimulated, it closes its sodium gate. Remember, this is unique in the eyes. Everything else we've studied, you open the sodium gate in response to stimulation. Now that that sodium gate is closed, the photoreceptor stops secreting neurotransmitter. So this means you no longer have that inhibitory postsynaptic potential. And since you no longer have that, you're not inhibiting the bipolar cell. So now the bipolar cell has an action potential. So then it secretes neurotransmitter to cause the ganglion cell to have an action potential. And because this ganglion cell is having an action potential, you signal the brain. Okay. Now we're going to go into a little more detail of what's happening in the photoreceptor, of just how you open and close that sodium gate. And this is a molecule called rhodopsin. So the purple thing here is your rhodopsin. Then we also have this yellow thing inside of it, this kind of orangish thing here. This is retinol. Okay, have you ever heard that eating lots of carrots is good for your eyes? That's not completely true, but there is some truth to that. Retinol is made from vitamin A. So you have to have vitamin A in order to have retinol, and you have to have retinol in order for your eyes to work. If you lack vitamin A in your diet, then you will lack retinol. And if you lack retinol, your eyes can't see. So a lack of vitamin A can cause blindness. Well, the whole carrot idea is because carrots and other similar vegetables have beta-carotene. When you eat them, your body converts the beta-carotene to vitamin A, which you then convert to retinol, and then your eyes work. So eating extra carrots isn't going to give you supervision. But if you don't eat enough stuff with vitamin A, you can lose your sight. So let's go back to exactly what the rhodopsin and retinol do. Here the retinol is bent. So this is called cis retinol. Here you can see the retinol is straight. This is called trans retinol. When light hits your eyes, it changes the retinol. It goes from cis to trans. So this is the light rays. These lines represent light. So when light hits your eyes, it causes the cis retinol to change to trans retinol. When that happens, they dissociate. Notice now here is the trans retinol, and here is our rhodopsin. They're no longer together. They've dissociated. This is called bleaching. The trans retinol dissociates from the rhodopsin. And remember, this happened because of the light rays. The light rays caused this. So this is in response to light. This is what closes the sodium gate. So this process right here, the bleaching, is what does this. Light enters your eye. The light causes the cis retinol to convert to trans retinol. The trans retinol then dissociates from the rhodopsin, and that causes the sodium gate to close. Now that the sodium gate is closed, you stop secreting inhibitory neurotransmitter from the photoreceptor. So the bipolar cell has an action potential, the ganglion cell has an action potential, and you send a signal to the brain. Then, after bleaching, you have to reset before you can send another signal to the brain. So now, this represents an ATP. So now you use ATP, and notice we're going to rebend that retinol. So the ATP converts the transretinol back to cis. And then it reassociates with the rhodopsin. Once this happens, you are ready to send the signal to the brain again. So signals from your eyes to your brain are not continuous. It's more like a strobe light, but the strobe light happens so fast you don't notice. So it is not one continuous signal to your brain. It is a series of individual signals. But it happens so fast, we don't notice it as individual signals. I'm sorry, we see it as continuous. It's like the old-fashioned cartoons where artists would actually sit and draw page after page after page, where each page was just slightly different, and then you flip through those pages so fast that it looks like continuous movement, but it's actually a series of individual ones. So now, once you get to this point, then you are ready to repeat. This idea is going to come up important with the night vision. So we're going to look at rods and cones, and then we're going to come back to this idea of that retinol converting from cis to trans and then back again as how you see. So now let's take a minute and look at rods and cones. These are your two types of photoreceptors. Rods only see in black and white, in shades of gray. They are your most abundant. And they are all over your retina. And these are what give you night vision. So when it's dark out, but you can still kind of see, you're using your rods. Cones are what give you your color vision. You don't have nearly as many cones, and they are concentrated in your fovea centralis. So they're not spread all over the retina like the rods are. They're mostly in that fovea centralis of the retina. Remember, that's the spot on the very back of the retina, just below the optic disc. They also give you your sharp, clear vision. They are what you use to read. So they give you color vision, and they're able to make clear images of small objects. They only work in bright light. So you can't really use your cones in the dark. Dark is for rods. Now we're going to think about, say it's a bright sunny afternoon and you go to the movies and you go from this bright sunny day into a dark movie theater. At first you can't see, but after you've been in there for a while, your eyes adjust and you do see. That process is called dark adaptation. So dark adaptation is when your eyes adjust to the dark. And at first you can't see. But then after a few minutes you can. Let's say that we're looking at a rod here. So we're looking at the rhodopsin and the retinol in a rod. Remember that process you cycle through. So here is where you are ready to see. Right, this is one all set up, ready to go. Here's the light rays. This is where you do that conversion. So the light rays cause the conversion. You bleach. And bleaching is what closes the sodium gate. And when you close the sodium gate, you signal the brain. Right, so remember we just talked about that up here. When the sodium gate closes during the bleaching, that is when the photoreceptor stops secreting inhibitory neurotransmitter, so then the bipolar and ganglion cells can have their action potentials and they signal the brain. Okay, then you have to regenerate. This is where you use ATP to convert that retinol from trans back to cis, and then you're ready to go again. In rods, this regeneration process is slow. So rods are slow to regenerate. So remember that strobe light? In rods, it takes a long time between flashes. Rods are like a slow strobe light. So when you are in bright light, that bright sunny afternoon, this overwhelms the rods. They are bleached out faster than they can regenerate. So your rods are basically useless in bright light. They can't keep up. So when you first go into the dark, so now you enter the dark. Now remember, cones only work in bright light. So as soon as you enter the dark, your cones don't work. And your rods are bleached out. So for those first couple minutes, when you go from bright light into the dark, neither photoreceptor is working. So this is where you're kind of stumbling around. You've got your hands out in front of you. You're trying to feel your way through, find your seat, not spill your popcorn, get into the movie theater. But then after a few minutes, your rods get caught up. And now that you're in dim light instead of bright light, they are able to keep up. So now you can see just fine in the movie theater once your eyes have adjusted. Okay, so now think about pirates. They have an eye patch, right? Everybody thinks they have an eye patch because they lost an eye in a sword fight. That's not the case. The purpose of the eye patch is for dark adaptation. When you are above deck, it's very bright and sunny. When you go below deck, it's dark. You don't want to have to waste time waiting for your eyes to adjust every time you go below deck. So the patch covers one eye. This prevents the rods in that eye from bleaching out. Then when you go below deck and it's dark, you just flip the patch to the other eye. Then your eye that is bleached out is covered up and you're not trying to use it anyway. And now you use the eye that is already adjusted to the dark. So you can see right away. So the idea is not that you lost an eye in the sword fight. It's that you use this eye when you're in the bright, above deck, and you use this eye when you're in the dark, below deck, and you don't have to wait that few minutes for your eyes to adjust.